We know that DNA is passed on from parents to children and holds all our genetic information. But how does that string of A's, T's, C's, and G's make us who we are? It's because of short segments of DNA called genes, which code for proteins, and it's those proteins that give us our hair and skin its color, make our muscles work, control the chemical reactions that break down some molecules and build others, and let things in and out of our cell. The process of making these proteins is called protein synthesis, and it's done in two steps: transcription and translation. Back in Chapter 2, you learned that proteins are polymers, made up of monomers called amino acids. A long chain of amino acids is called a polypeptide, and one or more polypeptides may be wound together to form a single protein. The instructions to put those amino acids in order are coded for by mRNA. mRNA is called the genetic code because the order of nitrogenous bases in mRNA code for specific amino acids that make up the protein. mRNA is like the master blueprint because it is created in the nucleus from the DNA template and carries the instructions from the DNA out to where the protein is actually constructed on the ribosome. The mRNA code is read by the ribosomes in groups of 3 bases called codons. Each codon codes for one amino acid, which gets added to the polypeptide chain. There are 4 RNA bases, A, C, G, and U, so there are 64 different combinations of possible codons. However, there's only 20 amino acids. The genetic code is redundant, meaning that several codons code for the same amino acid. For example, 3 different combinations of A, C, G, and U code for stop, telling the ribosome where the end of the polypeptide chain is. The genetic code is also universal, meaning that the same mRNA bases code for the same amino acids regardless of the organism. This allows for some pretty cool possibilities. Monsanto, a local biotechnology company, has put genes from one plant into another, allowing the protein-making machinery from one organism to use the instructions from another to make proteins. Using this technology, we've been able to make worm cells glow like fireflies, plants that resist disease and weed killer, and pigs to make the human protein insulin. Just a little teaser. We'll learn more about that in our biotechnology chapter. It's also what allows viruses to invade our cell. Viruses inject their genetic material and hijack our cellular machinery to make their proteins, and they essentially take over our cells. Every protein has 2 things in common. It must start with a start codon and end with a stop codon. This is what tells the ribosome where to start and stop. There is only one start codon, which also codes for the amino acid methionine, if it's located after the start codon. There's also 3 different stop codons. It's not necessary that you memorize these, but you'll end up knowing the start codon by the end of the chapter. So here's how the message coded for in the DNA ultimately gets made into a protein. The process has 2 steps: transcription, which makes the mRNA blueprint, and translation, which takes that blueprint and uses it to build the protein at the ribosome. Transcription is actually pretty similar to DNA replication. First, DNA is unwound. RNA polymerase is the enzyme that unwinds the DNA and also adds the RNA nucleotides. Just like DNA polymerase in replication, RNA polymerase adds nucleotides from 5' to 3'. So the 3' to 5' strand is called the template strand. The mRNA that's created from DNA is always complementary to the 3' to 5' DNA strand. RNA polymerase needs a signal about where to begin adding the RNA bases. In replication, DNA polymerase uses a primer to signal where it should begin. In transcription, a sequence called the promoter signals the RNA polymerase where to start. A sequence called the terminator tells the RNA polymerase where to stop. Once the mRNA has been created in the nucleus, it goes out to the ribosome, where the message is translated into a sequence of amino acids. This process is called translation. tRNA plays an important role in translation. You can see that the tRNA actually looks like a T. The top part of the T contains a 3-base sequence that's complementary to an mRNA codon. This 3-base sequence is called an anticodon. The other part of the tRNA has an amino acid attached to it. During translation, mRNA attaches to the ribosome, and as it moves across the ribosome, tRNAs are recruited to bind with the mRNA codons and attach their amino acid to the polypeptide. This picture shows a good summary of the entire process of protein synthesis. A small strand of DNA unwinds in the nucleus, and RNA polymerase adds RNA bases to make mRNA. The mRNA then leaves the nucleus and attaches to a ribosome. The ribosome then recruits appropriate tRNAs to bind their anticodons to the codons found on the mRNA. As the tRNA binds to the mRNA, it attaches its amino acid to the polypeptide chain. And this continues until the stop codon is reached. This picture of the whole process is good to help you explain how a protein is made. So don't forget to go back and complete the quiz on Edline. So this is in the assignments folder, just like always. And here is a preview of the questions from the quiz.